Good morning. My name is Abby, and on behalf of my group, I'd like to start a conversation today about schizophrenia and the genetics behind it. So let's start by taking a look at this picture. It was drawn by the artist Sue Morgan, who has schizophrenia herself, and in the picture, she's trying to portray what it's like to have schizophrenia. So your brain is kind of scattered, and this is one of the positive symptoms of schizophrenia. There are a lot of other symptoms, including delusions and incoherent speech, and also just generally odd behavior. And there are also negative symptoms of schizophrenia, which are the lack of normal behaviors, such as lack of motivation, lack of normal emotions, and being catatonic. So a person with schizophrenia can have these symptoms in any different combination. And the diversity in the symptoms of schizophrenia led scientists to at first question whether schizophrenia was even a disorder, because it's so diverse and people have such different things going on. Then, once they accepted that it was a disorder, they thought, okay, it's probably based on a single gene, like a lot of other disorders that we've researched. But as more research was done, they realized that it was actually polygenic and highly heritable. So there's a lot going on with schizophrenia, and it's becoming more complex as more research is done on it. One thing that scientists do know about schizophrenia is that neurotransmitters are involved, and specifically an increased activation of dopamine receptors and decreased activation of glutamate receptors. So the most effective treatment that we have currently targets these dopamine receptors in the form of antipsychotics. These antipsychotics are highly effective in treating the symptoms of schizophrenia, such as hallucinations, but they're not treating the core cause of the disorder, and they have severe side effects. So one way scientists are researching more about schizophrenia is by doing genome-wide association studies, which we'll call GWAS. And a recent breakthrough in the schizophrenia community found 108 new gene loci that are corresponding with schizophrenia. So they're shown on this Manhattan plot. So if someone has a variant in one of these 108 genetic regions, they're highly likely to have schizophrenia, which is completely new for schizophrenia research. So that study was done by the Psychiatric Genetics Consortium, and this same group also compiled GWAS data from 60 other institutions, and they wanted to map the genetic mutations to where they were actually occurring in the body. So they found them in two specific regions: the brain, which we would expect because that's where dopamine receptors are, but also in cells that are important to immune function. So now scientists are also speculating there's a connection between immunity and schizophrenia. So all of the things from GWAS studies can be used as targeted drug therapies, which is one option for drug development. The traditional option for drug development has been just developing antipsychotics that are treating the symptoms, but now organizations like the NIMH are exclusively funding drug therapies that are targeting genetic regions, and they're not even looking at antipsychotics anymore because they're just treating the symptoms. So to test these therapies, as we talked about in class today, they use mouse models. And to start a mouse model, it comes in two steps. First, you have to induce a change in the mouse that's going to do something like schizophrenia does, and then you have to measure the effects. So observe the behavior of the mice to see if they have schizophrenia. The first model that they came up with for mice is called the classical model, and they did this by administering amphetamine to the mice, which ramps up dopamine levels, and the symptoms that they were able to observe are hyperactivity or passive avoidance, which are similar to human symptoms of schizophrenia. And this was based on the fact that dopamine receptors are highly active, and also if humans take amphetamine, it ramps up dopamine levels. An additional mouse model that has been invented is by damaging the hippocampus in newborn mice. So by doing this, they had no initial symptoms of schizophrenia, but actually later in life, they seemed to be schizophrenic because they were socially withdrawn. So this is now suggesting to scientists that schizophrenia is not only a genetic disorder, but it's also developmental. So again, increasing complexity with schizophrenia as we're researching more about it. A third mouse model that the pharmaceutical industry has come up with is trying to standardize the mouse model because they need something that's highly reproducible. So if they're testing their drugs on it, they know that it's always having the same effect. And the way that they are doing this is by altering cell development in pregnant mice, and then in their offspring, they're observing schizophrenia symptoms such as ventricular enlargement that you can see here, and also cognitive changes that are similar to human schizophrenic symptoms. But drug development with mice is problematic because mice are not humans. So schizophrenia is polygenic, so if you induce one change in a mouse, it's not necessarily going to result in the schizophrenia phenotype. It also has low penetrance, so if you're inducing a change that you think should give the mouse schizophrenia, it might not always work. And mice also present with ambiguous phenotypes. So how can we be sure that a mouse has schizophrenia? We don't really know. So a lot of people question the validity of mouse models because we don't know, does a mouse have schizophrenia? Can you really tell? Another problem with drug development is that we just keep making different versions of antipsychotics, which is called the problem of Me Too drugs. Because the antipsychotics that we're using today are actually just as effective as the ones that have been used for 60 years. So we're kind of at a dead end there. Shifting gears a little bit, there's also a connection between schizophrenia and creativity. If you think about artists in history like Van Gogh, he was crazy, but he was also super artistic. So people want to see if there's a genetic connection between these two things. And a recent study that was done at King's College London actually found that the polygenic risk scores for schizophrenia can predict creativity. So the same genetic mutations that are predicting schizophrenia can also predict how creative a person is. Sue Morgan is a perfect example of this because she's artistic and she also has schizophrenia. So this calls into question, if we treat her schizophrenia, are we also treating her creativity and limiting that creativity for the world? But before we answer this question, we have to think about what is the best treatment for her right now. So we have to go through all of the GWAS studies, the GWAS data that we have, and that brings us to our propositions.
So here are our propositions.